In this class, we will talk about the different experiments that allow us to know how genes were located on chromosomes. So remember, key here that I want that you remember is that genes are located on chromosomes. And the first part of this lecture, we will talk about how many scientists start trying to test the Mendel's genetic model. And so there was a lot of activity trying to validate or disprove Mendel findings because definitely we know that it's clear that these ratios take place, but there was no mechanism explaining why these ratios take place. And to understand how the mechanism occur, the first thing that we need to understand is the chromosomal theory of inheritance. So we'll talk about the work of these two scientists and how they have careful observation of a process called meiosis. And then we will focus on the work of this scientist that is very important, that is Thomas Morgan and his experiments on fruit flies, that he was able to find that using fruit flies, and in particular the eye color, he was able to find that genes were located on chromosomes. Finally, at the end, I will talk about another piece of information that it was fundamental for Thomas Morgan to understand that genes were located on chromosomes, and it was the discovery of the sex chromosomes. If you remember that there are X and Y chromosomes, these scientists, using the karyotypes that you see here, she found that chromosomes of males and females in the X and Y, as you can see here, they are different. And that was a key piece of information for Morgan to find that genes were located on chromosomes. So let's start. So now let's start talking about this first point. Okay, so to start the class, I told you that after the rediscovery of Mendel, there was a huge activity in the scientific world to try to understand not only Mendel results, but in particular the mechanisms. Yeah, so we know that there were Punnett squares, if you remember, that allow us to make crosses and to predict genotypes and phenotypes. Yeah, for example, here you will get a specific pattern. You will get 1 to 1, and you will get a ratio of 3 to 1. But why we get these patterns? How do we explain this? And many scientists are trying to prove and disapprove Mendel's ideas. And in particular, there was an organism that we will talk today that, in addition to the pea plants, we start using the fruit flies. And in particular, we start using here, we will talk about the eyes of a fruit fly, that it became a very important model to understand genetics and to understand how genes were located on chromosomes. And in particular, it was an undergraduate student that it seems that he was the one who found a mutation that it was key to understand why genes are located on chromosomes. So remember, Mendel created this fantastic model that it was used by many scientists for many years. However, it was only had like hypotheses, but we need to explain those results and we need to try to find the mechanism. And we had Punnett squares, that they were a wonderful tool to predict the genotypes and the phenotypes. However, we still don't know the mechanisms. And to do that, many scientists start trying to test to try to validate and try to disapprove Mendel findings. And to do that, they start trying to use many different organisms in addition to pea plants. And they found that not only Mendel was right, but they use multiple tools and they were able to find that genes were located on chromosome, and that was fundamental to try to understand that. And in particular, they will start trying to use not only Drosophila, but they start using model organisms and mutations. So if you see here, this is an example of a mutation. In the wing of the fly here, you have a wild type that is considered a normal phenotype. However, here you have a fly with a curly wing, and this is very important because this is an example of a mutation, and after this time we start studying mutation to try to understand genetics. So now that we talk about how people were very interested in testing the genetic model of Mendel, one way to test that model was to study chromosomes and propose this chromosomal theory of inheritance. And we will focus on the work of these two scientists that are in this picture, using fertilization and meiosis. So one of the key components to try to understand how genes were located on chromosomes is this finding, is the chromosomal theory of inheritance. And as I said before, it was proposed by Sutton and Boveri. And how they did it? Because they have independently, they carefully observed the process called meiosis, and we will talk about that in a second, and I will show you a video of that. And what they did is, you remember Mendel's work that he found some hereditary determinants. So what they propose is that when they study meiosis, propose the chromosome are composed of these hereditary determinants that now we call genes. But they proposed more things. So they proposed that the chromosomes carry these hereditary units and also organize them. So they are organized in the chromosome. So for example, if you have here one of these chromosomes that you observe here, so they found, do you see this green seed? Yeah, they should be located in one specific region of the chromosome. And do you see that this is not smooth anymore? Maybe this should be located in another section of the chromosomes. So just to recap, remember that these two scientists proposed the chromosomal theory based on their observation of this process called meiosis. And then they state that chromosomes are composed of Mendel hereditary determinants that now we call genes, and that this theory was proposed independently, and they found that the chromosomes were not only the carriers of these hereditary unions, but they were organized in different locations of the chromosomes. Before that I start talking more about the chromosomal theory, I want to tell you why they were able to propose the chromosomal theory. And one of the key things is that now, in addition to have one model organism that Mendel used, that it was the pea plants, now they start using multiple model organisms like fruit flies. And we will talk about that. And in addition to that, Sutton and Boveri used other model organisms, but that was not the only change in approach that they did. Now there was a huge advancement of microscopy and related techniques. So in the beginning of the course we talk about homunculus and that there was not enough resolution. However, now they were able to have a better resolution and they were able to identify chromosomes in this structure, and that was very important. And one key piece of information or key evidence is that as microscopy progressed so fast in this time, scientists were able to study the process of cell division, and in particular, they were able to study meiosis. And there is a new field, so that is called cytology. And cytology is the study of individual cells. So while cytology study one cell, histology study the entire tissue. And in the 19th century, this field started growing really fast. There were many discoveries that they were done at that time because the light compound microscopes were not anymore producing distorted images, and this will allow to test and produce new discoveries. And this was fundamental. So remember, in the 19th century, there was a field in vogue that it was cytology, because as microscope started improving their images, there were many discoveries made at that time. So they went from this type of images to this type of images. So the images were not distorted anymore. And this allowed to validate evidence for new theories. So one of the examples of these cytologists was Theodor Boveri, one of the persons that proposed the chromosomal theory as we watched in the previous slides. And this German, as I said, he was a leading cytologist, and he used as a model organism this organism that is called the eggs of the sea urchin. And why? Because they are a great system to study the cell nucleus after fertilization. And I will show you that in a second. So although right now we have stainings that allow us to see the nucleus in a very easy way, and that allow us to see the study the process of meiosis, the sea urchins are particularly good to study this process of fertilization, and the cells are relatively big and they are very easy to study in the microscope. So this is why he picked this model organism. That, remember, I might ask you in the exam that he used eggs of sea urchin to study cell nucleus and chromosomes during meiosis and after fertilization. Maybe you have never seen the eggs of a sea urchin. So I'm going to show you a video, and I'm going to show you a person that extract the eggs, and so you understand why that was a good model to study this process.
You will now see the process of fertilization in vivo thanks to the sea urchins provided for you in this laboratory. Some of the developmental life stages of the following young larvae can be viewed using the following procedure. Place the sea urchins in specimen dishes containing enough seawater. Inject 0.5 milliliters of 0.5 molar potassium chloride on each side of the mouth located on the oral side of the sea urchin. You do not have to inject them too deep. Keep in mind that sea urchins have a nervous system and they are capable of feeling pain, so please handle them properly. The potassium chloride allows for the release of gametes from the gonopores located on the umbral side of the urchin. If a white fluid is released, then you will know it is sperm. Collect the fluid with a pipette and store it on ice in a plastic tube. When finished, place the pipette in biohazardous waste. Keeping the sperm on ice will slow down metabolic activities and keep the sperm in good condition for the upcoming fertilization process. If a reddish fluid is released, then you will be dealing with eggs. Collect the fluid with a pipette and place it in a 100 milliliter beaker containing some seawater. When finished, place the pipette in biohazardous waste. Place 1 drop of seawater containing eggs on a glass depression slide. You do not need to put a cover slip on it. Now you're able to look at an egg under the microscope. Observe its different parts, such as the cell membrane, cytoplasm, and nucleus. Now dilute the sperm in seawater by placing 2 drops of sperm and 10 milliliters of seawater in a small beaker. Place 1 drop of the sperm solution onto your depression slide containing the eggs. You can now view the fertilization process under the microscope. Observe the sperm and the egg. Cell division starts with the first cleavage where the zygote splits into two cells approximately 90 minutes after fertilization. After another hour, the second cell division starts in 4 cells and division continues to form 8 cells, 16, and on to the 128 cell blastulus stage, approximately 1 day after the egg was fertilized. When you are finished with the lab, put the sea urchins in the seawater container labeled used. The used sea urchins will be returned to the sea.
Okay, so as you saw, sea urchins' eggs were ideally suited for the study that Boveri was planning because they are easy to fertilize in the lab, and other models organisms they were much difficult. In addition, if you see have a quick time frame for larval development, so with plants like the ones that Mendel used, this would have been very difficult. And for example, with fruit fly, this also is also very difficult, especially at that time. And something that is not that they don't talk in the video that I want that you remember is that Boveri he not only did observations, but he also did experiments. That is fundamental to study biology because you can prove, make hypotheses, and then test it using experiments. And although describing the phenomena is important, doing experiment is key to test hypotheses.